So, on behalf of Mahavet, I welcome all the participants for this CVE named Refreshing Course on Ruminant Reproduction. Under this series, we are going to conduct series of lectures which will be focused on the ruminant reproduction. So today we will be starting in this series from first topic about basics of ruminant reproduction, in which we will be covering detailed overview of the normal estrous cycle, its neurohormonal regulation, artificial insemination, and fertilization. So for this topic, we have a very eminent and a respected speaker, respected Dr. N.M. Markandeya sir, who is retired Associate Dean from Parbhani Veterinary College, Mapsu. At Animal and Fishery Sciences University. So I welcome Dr. Markandeya sir for this first topic of this CVE series initiated by Mahavet. So this language of communication will be English because many of the participants, field vets as well as budding vets, have joined all across the nation. So I welcome respected Markandeya sir to start his video and he can start his session. So Dr. Akshay, once sir will start, you can share the screen as per his need.
Yes, sir.
Share the PPT.
Okay, sir.
Should I start?
Yes, sir.
Yeah, thank you. So, good evening, all professional friends. It's my pleasure to be with you for this evening. Maharashtra State Gazetted Officers Union has launched this continuous education program, and I was asked to open the session with basics of farm animal reproduction. Friends, training is a part of professional life. Regularly, one should involve in the training, and also one should update the knowledge for its implementation in clinical cases. We are attending clinical cases where applicability of basic knowledge is most important one. The timing which we have selected is most appropriate because everyone is really comfortable at this evening hours, and also it is possible to discuss in length without limitation of time. I'm also at my home, very comfortable, and therefore the topic I would like to cover, I will finish it within 90 minutes, and thereafter please join with me for your questions. As far as this topic is considered, reproduction is most important because we have to go for such a type of cases which are very, very delicate, which are very, very important for the farmer breeders. If there is a clinical case of a fever, indigestion, tympani, we can easily diagnose it and we can give the treatment. But in case of reproduction, even after exploring many type of therapies, response from the animal is not as per our expectation. And this is particularly true when we deal with subclinical endometritis like things, type of estrus detection, so on. So this particular topic is very important, and let us discuss about the basics so that in the very next topic, we will be able to discuss clinical type of things. May I request to have the first slide? I think this is the last slide. Please. That's right. Let us start with this. In case of males, reproduction is a continuous type of activity. Continuous sperm production, continuous type of services. Once breeding starts, this particular phenomenon ends with senility. But in case of females, remember, it's a cyclic and phasic. Estrus cycle, gestation, followed by postpartum phase, again resumption of cyclicity, cycles, conception, gestation, and so on. So this is cyclic and phasic, totally different type of activity in males and the females as far as this mammalian reproduction is considered. Friends, this particular system of reproduction, which is working in males and the females in a very different manner, has 5 characteristic things. Please remember that why we are not getting our expected results, it is only because these type of important characteristic things of the reproductive system. Number 1, separate type of organ system in case of males and the female. This is the only system in the body which is different in males and the females. Otherwise, digestive system, nervous system, vascular system, any type of system is exactly the same. Even our musculoskeletal system, everything is the same. This is the only physiological activity in the body which is dependent upon normalcy and regularity of other body systems, other body processes. If physiology of other body systems and functioning is normal, then only animal can perform best as per the reproduction is considered. Third important thing, in case of bitches, we know that even if we remove the entire reproductive system, animal can survive. So survivalence is not related with this particular system. It's not required for the life, for survival of the life, but it is most essential for the life because this is related with the reproduction. This is the only system which is directly related with the reproduction as production, and therefore unless there is a production, we cannot maintain the animals. So in that manner, it is required. But as per a physiology of the life is considered, this is not at all required. This is the only system they are having ovaries, fallopian tubes, uterus, cervix, everything. These organs, they cannot work for their own accord. Neurohormonal control is required. So nervous system and the hormonal system is playing role for this functioning of the reproductive system. Important thing is that hormones, they play the role of wireless system, and the nervous system is playing the role as electrical system. So electrical system and wireless system, neuroendocrine control is most important. This is the only system in the body which will not start its functioning right at the time of birth, but it will start in the latter part of the life once there is a growth and the development of other bodily systems which are oily, essential, and vital for the life. Those systems will develop first, and then only the reproductive system will start its functioning. This is very, very important. 6th point, you can very well adjust with that of the production, that it is the only system which is directly related with the production. Otherwise, there will not be any chance of production in absence of reproduction. So friends, this is the characteristic, and therefore reproductive system always behaves in a vagabond manner. It has no regular path in case of farm animals. We are observing it. We are regularly observing it. Smooth reproduction that we never expect. 21 calvings in a cow, 19 calvings in a buffalo. This indicates, yes, everything was smooth throughout the life. But when it comes to only 5 lactations, 6 lactations, 7 lactations in cows and buffaloes, we understand that there was problem in between, and therefore the reproduction has stopped. Next. So the next. Friend, we'll go to next slide. Hello.
Dr. Akshay,
Can you give me this? Okay, that's right. Today's topic, we are going to deal with the breeding and reproduction. Breeding is related with the breeding policy, which is linked with the reproduction. We are going to discuss both fertility and fecundity, where we will be covering farm animals, cows, buffalo, sheep, and goats, particularly growth and the puberty, because that is a part of reproduction, followed by estrus, its detection, its confirmation, and the final artificial insemination and conception. Once animal becomes pregnant, then everything will go smooth as we expect in case of the animals. Next. Right. In case of animals, particularly say for farm animals and that too for cows and buffaloes, large ruminants, this is what we expect from the cows and the buffaloes. On an average, if we consider crossbreds and our native stock, everything, for that purpose, first calving should have been between 24 to 48 months of the age. If this is 24 to 28 months, it implies that animals should become pregnant at the age of 15 months to 19 months. If we expect pregnancy, first pregnancy by 15 to 19 months, that means that heifer should come into estrus by 12th month, so that with the first 2 irregular cycles, she will settle in the 15th month of age, and then she will produce the first calf by 24 to 28. In case of buffaloes, same thing is expected with 34 months as a target. 32, 34, 36, we can have our own targets. Calving interval, this is international phenomenon. We have accepted, largely we have accepted that 365 days should be the intercalving interval in case of cows, whereas in case of buffaloes, let it be 420. That will suffice the purpose, because gestation period is more, number one, and number two, we expect milk up to 10th month in case of buffaloes. In case of cows, it is up to 9 months. In case of buffaloes, up to 10 months, and therefore 10 months gestation, 10 months lactation. So that should match, and therefore 420 days intercalving interval. Days open, we are expecting less than 100 days. That means we are expecting conception by 81st day in case of cows, and we are expecting it in case of buffaloes by 111th day in case of buffalo. So that is less than 120 days. Pregnancy rate is indicate the total number of animals which should become pregnant on a farm must be 90%. So out of 10 cows, 9 cows must become pregnant yearly. When the farm is of 10 animals, 9 cows should become pregnant. The same is true in case of buffaloes. Calving rate of the pregnant, 90%, 95% cows should carry the total pregnancy up to 95%, means calving should be 95%. In buffaloes, problems are more, and many buffaloes, they have gestational problems. So at least 90% calving is expected in case of buffaloes. And on the basis of reproduction, on the basis of poor reproduction, only less than 8% animals should be culled from the farm. Means culling is not killing. Culling means removal from the breeding policy. 8%, less than 8% should go out on the basis of reproduction. In case of buffaloes, less than 6% we expect in case of buffaloes. See, in case of buffaloes, we expect less because buffalo reproduction is more precious than that of the cows. Next important. Next slide, please. That's right. The first and the foremost part which is ignored in the reproduction is the growth. Growth is part of reproduction. We cannot ignore growth at all. And the growth starts during prenatal life. 7th, 8th, 9th month of gestation. This is indicating that calf is -3, -2, -1 month old. And during the gestation period itself, the maximum growth takes place. And we expect highest birth weight in case of calves. Maximum birth weight is more important because healthy calves definitely will have better growth rate in the life. Higher birth weight is expected. Many people, they feel that if the birth weight is higher, it will lead to dystocia problem. Forget about this. This total birth weight is controlled by the mother. It is not controlled by the sire. It is controlled by the dam, and therefore always dam will control the birth weight, and accordingly sizable type of the fetus will be delivered out. So no need to worry about the higher birth weight. We have observed such a type of higher birth weights in case of Murrah buffaloes, crossbred cows, and now these figures are moving 50 plus. So no problem as per the birth weight is considered. Higher birth weight is expected. Postnatal growth. This totally depends upon birth weight. Higher the birth weight, maximum is the growth rate. Please remember, higher is the birth weight, maximum is the growth rate, and therefore the calfhood stage should culminate early. Animal, that heifer should become mature early. She should express the first estrus early, and for that purpose, the growth rate must be constant. It must be higher. It must be increasing, and it should not be interrupted by any type of factors. So, in smooth reproduction, this is possible. This is possible. This growth rate needs to be controlled, and it needs to be controlled by weight only. Weight measurement is important part in animal reproduction. Those who are ignoring animal reproduction, this recording of the growth rate in case of the heifers, they are likely to suffer with reproductive problems of their farm animals, and therefore monitoring the growth rate is important. Onset of puberty is more related to development than age. So it is interaction of age and the body weight. Interaction of both body weight, skeletal weight, and the age, calendar age. Friends, age is also important. Let the heifer grow up to 9 months, 10 months. Skeletal weight must be 250 kg in case of cow heifers. In case of buffalo heifers, it is 275 kg. In case of goats, it is expected to be 21 kg. And in case of sheep also, it is something like 20 kg. If this particular body weight is achieved, animal is able to express the first estrus, and pubertal estrus expression. It is very, very important to understand that. Number one, growth hormone level stabilizes. First instance, when the growth hormone level declines, it declines. This is the first signal for puberty, number one. Number two, metabolic fuels in the body related with the growth factors, and particularly the insulin growth factors, IGF-1, IGF-2, when these factors are stabilizing, when growth hormone has its influence on glucose level, very important, insulin and glucagon balancing, when it is rational, and most important thing is that when there is external stimulus, and this stimulus, it is environmental factors are playing role in this, like pineal body is there, followed by daylight, olfactory sensations. So these things, they should unite and they should stimulate hypothalamus for initiation of puberty. Then only puberty will start. Otherwise, many helpers will have metabolic fuel levels, but they may not start the puberty only because external factors are not playing the role. Biostimulus is one of them. So please remember that when there is a synchronization of all these factors, the first stress is expressed in case of the animals. Generally, we consider that 40% of the mature body weight is necessary for initiation of puberty. And puberty indicates first stress. Once animal achieves 40% of its mature weight, animal will come into the stress. First stress is always a mad desire. The word is very important, and that is called as the mad desire. Mad desire means animal will express secondary sexual characters and will not have proper reproductive sequencing, and therefore the first stress, it is just to be ignored. It is to be recorded and then it is to be ignored. Once there is an initiation of follicular growth and expression of the first stress, this particular physiological event will lead to sequencing of the events, stabilization of levels of hormones, particularly steroids, their expression on higher centers of the brain, and then animal will respond better in the next subsequent cycles for expression of the stress. So once there is a pubertal stress, many helpers, they are taking 40 days, 45 days to express the second. From second to third, it is 35 days. From third to fourth, it is something like 27 days. And then the cycle is recovered. In other animals, in other heifers, their growth rate is very fast. Remember, with the second and the third cycle itself, there is a regularity in the stress. And once there is establishment of the regularity, definitely this animal is fit for artificial insemination. Next. So we have discussed that this particular stress of the first time in life, this is indicating that the reproductive system is totally prepared for the reproductive capabilities. But 2 cycles, 3 cycles, which are required for the regularization of the cycle, that much period is given to reproductive system to develop. And once this reproductive system is developed with 2, 3, 4 cycles, 60 days, 80 days, 100 days, this particular time phase of 3, 4 cycles is required for maturity. That particular period is important. 4 months, 5 months period is period required for maturity. It is a maturity of reproductive system. It is maturity of reproductive organs, and it is maturity in terms of sequential expression of reproductive events. So once animal is mature, then only animal is able to bear the reproductive capabilities, that means the pregnancy. Otherwise, pubertal stress, first 2, 3 cycles are not at all expected for covering that particular animal and breeding of that particular animal. All field veterinarians are requested that with the first pubertal stress, it is our bounden duty. Please remember, whenever we are in touch with the professional dairy farmers, it is our bounden duty to attempt parental examination. Pubertal animal, parental examination is mandatory. That will signify, number one, normalcy of each part of the reproductive system, ovaries, fallopian tubes, uterus, cervix, everything we can palpate. Number two, it will assess the size of the pelvic girdle. Number two. Number three, this will assess that whether with all probabilities, this particular animal will grow and develop and carry the ability to carry the pregnancy. So all these things can be assessed with the first rectal examination. On first rectal examination, if ovaries are defective, if cervical problems are severe, if uterine problems are severe, we can indicate that particular owner that this animal is going to face some or other type of problem. But for confirmation of normalcy, our examination is necessary. Now, before going to cyclicity of these animals, it is important to know this particular picture, and also it is important to know the glands which are involved in secretion of the hormones which are directly related with the reproduction. Hypothalamus, master gland, is brain, higher brain centers, or it is situated at the base of the brain, and this particular hypothalamus is controlling the reproduction. All signals, there are dozen and more signals which are released from the hypothalamus for all types of hormones, and there are a dozen and more all types of inhibitory factors or the hormones from the hypothalamus to control the release, to inhibit the release of these hormones from pituitary gland. This is the site where oxytocin is produced and it is ported to the posterior lobe of pituitary. So hypothalamus has an important function in animal reproduction, and the most important thing is that the hormones or the signals of hypothalamus are chemical in nature, very safe for use. As far as the GNR is considered and its utility in animal reproduction is considered. This particular part is important. The next important gland is pituitary, anterior lobe and the posterior lobe. Anterior lobe is related with the three non-reproductive hormones and three reproductive hormones, whereas posterior lobe is related with the vasopressin and oxytocin, these two sets. Somatotropic hormone, STH, that is the growth hormone. Number two, it is the follicle stimulating hormone. Number three, luteinizing hormone. This is ACTH, TSH. These are the hormones from anterior pituitary. Anterior FSH, LH, and prolactin, these are directly related with the reproduction, whereas STH, TSH, and ACTH, these are non-reproductive type of the hormone. They have no direct function. They are related with the reproduction, but there is no direct. Unless somatotropic levels, the hormone levels are declining, there is no initiation of estrous cyclicity. Please remember, the same thing is true in case of anestrous cases. How anestrous animals are coming into estrus, this is dependent upon the growth hormone. If this growth hormone should increase, and then it should stabilize, then only animal will come into estrus. Second one is the thyroid function, thyroxine. T3, T4 levels are important, and they are indicators of metabolic processes in the body. Controlling the total metabolism, thermal regulation, this is with T3 and T4. And the last one is the stress hormone, that is ACTH. Comfortable animals without any environmental nutritional management stress, are only expressed in the estrus. FSH is related with the follicular development. LH is related with the luteinization, and prolactin is related with the lactation that genes in them. Now, in this picture, you will also find placenta, a important part which is related with the gestation, and it is responsible for the production of the progesterone, conversion of progesterone into estrogen at the time of parturition, and there are many other functions for endocrine gland, that is placenta. Placenta as an endocrine gland. Ovaries are producing steroid hormones, estrogen and progesterone, and also near the kidneys, there are adrenal cortex, which are also important as far as the cortisol level is considered, as far as their role at the time of parturition is concerned. So this entire picture will indicate the type of the hormones which are related with the reproduction. Next one. Next. Right. Hormones are chemical messages secreted by one gland. Their action is on another gland, and target organ is different. Transportation is through blood. Proteins are required for carrying this hormone from one place to another place. And target organ is that organ where there are receptors. If there is a receptor, hormone will be recognized. FSH will be circulated throughout the blood, through all body parts, but only ovaries will recognize it because there are receptors to recognize FSH. Same thing is for the and same thing is for all other hormones. Peptide hormones, protein hormones, glycoprotein hormones, these are all the characteristics of anterior pituitary. So please remember that anterior pituitary has important role as far as these protein hormones are concerned. And whenever from market, when we are using the FSH-like hormone, LH-like hormone, these hormones are protein hormones, and their repeated use, it will lead to formation of antibodies against these hormones. And therefore, chemical hormones, chemical messengers are most important, and that we have to attempt. GnRH, decapeptide, oxytocin, carrying 8 amino acids. So these things we have to handle very delicately, and whenever we are using the hormones, we have to use them very judiciously. The next one. The next one. See, this part is more important for we people, that for how much time a hormone will act once it is released by the anterior pituitary. GnRH and oxytocin, you will find that this is less than 10 minutes. So 6 minutes to 8 minutes, that is maximum time in which the action is brought about. When you consider LH, it is nearly 45 minutes to 60 minutes action will occur. In case of FSH, you will find 4 hours, maximum 4 hours. That much is the time for which the action of FSH will continue. And when we go for human chorionic gonadotropins or equine chorionic gonadotropins, that is FSH like and LH like hormones, they have very long half-life, biological half-life, and therefore clinically those are used for their very long durable activity in case of animals. Next one. Next. In case of hormones, as a clinician, please remember that estrogen and progesterone, their role is different, but it is Ardhanari Nateshwar Roop. Ardhanari Nateshwar. One side male, one side female. Likewise, one side estrogen, another side progesterone. One side estrogen, another side progesterone. And same thing is true with the FSH and LH. FSH and LH, they have subunits alpha and beta. Their biological activity will depend upon union of these subunits. So whatever the basic material, what we call in local language as a masala, is always ready. Combination determines which type of hormone is getting secreted. Means see, all subunits are ready with anterior pituitary. Only combination will decide. Now this is FSH, that has to be released. Now there is LH, it has to be released. So only thing is that it is a combination and biological activation of that particular product which comes into the bloodstream. Next one. Next. Steroids. We have more important relation and we have more clinical applicability of this type of steroids. In case of steroids, cyclopentanophorhydrophenanthrene ring, this basic structure is most important, and you will find that always this particular structure is related with the steroidal type of the hormones. It is a conversion of one type into another. One hormone is converted into another hormone, and then its role will change. All types of hormones are prepared with the enzymatic type of actions. We are not going in detail about this enzymatic type of relations. Next one, please. We have to discuss a little more, and here you will find that there are synthetic hormones also. These synthetic hormones are more potent than natural one. Their half-life is also more in the blood, and therefore such a type of synthetic preparations are used clinically. For estrogens, long time we were using estrogens, estradiol benzoate, that we were using, but nowadays we have alternatives, and therefore estrogens are very rarely used in clinical type of cases. Of course, testosterone is required in case of the male animals, and we are using it in case of male animals, but synthetic preparations, they have more biological activity, more half-life, longer half-life, and therefore their use is more in clinical practice. Next one. Action of the steroid hormones, it is in the nucleus of the cell, target cell. Steroid hormone will be carried through blood. It will be taken up into the cytoplasm. It will act on nucleus of the cell, and then the action will be produced. So receptors are basically at the nuclear regions for action of steroid hormones. This is the part. Next, for peptide hormones, this action is always on the cell wall. So receptors are available on the cell wall. Cell wall itself will recognize the hormone, and the action will take place at the target. This is the concept, and this we should know. Next one. Next. This is the list, and I'm going to spare my PPT presentation to you all. It is only because this is informative, and whenever there you want to go in details, you can refer this type of PPTs. So for estrogen, the source is ovary. Estradiol 17 beta, that is the source. That is the hormone basic activity, and that is with ovary. Secondary sexual characters and estrous behavior, it is related with this. Estrogens are required for the growth and the development of female reproductive system. This is also important, but the system of mammary gland is also developed, particularly the tubular system is developed under the effect of estrogens. Same estrogen is converted to progesterone, and progesterone is coming from corpus luteum. Progesterone is directly related with the maintenance of the pregnancy, inhibition of uterine contractions, and this particular progesterone is important one because it is related with the fertility. Estrogen is not related with the fertility. Progesterone is related with the fertility. So please remember, higher the progesterone, higher is the fertility. As far as the male animals are considered, next one. Yes, Leydig cells and Sertoli cells, those are more important as far as the spermatogenesis is considered, and then testosterone production is considered. So, cortex, adrenal cortex will provide glucocorticoids, cortisol levels, and these cortisol levels, they play important role for parturition. So this is a different story for parturition and the late stage of pregnancy. But as I told, endocrine gland placenta is more important because it is producing FSH-like hormone, LH-like hormone in different species, in different way. All chorions, they are not producing same type of activity, but different type of activity. Placenta is producing estrogens, that is more important in almost all the farm animals. Relaxin in all the animals, and placental lactogens. This is also important, that is required for mammary growth and milk secretion. So these things are coming from placenta, and placenta also acts as the endocrine gland. The next one. Next. Yes, uterus. Local hormones are there. Neurotransmitters, we call them as prostaglandin. Very important one. And this is the local hormone. Prostaglandins are secreted by healthy endometrium. Only healthy endometrium secretes prostaglandin. This is the underlining sentence. It's a lipid in its structure, but this is required for luteolysis. It causes luteolysis. Causes luteolysis whenever animal is empty. When uterus is healthy and empty, PGF2 alpha will cause luteolysis. From liver, what we have discussed about the insulin-like factors, insulin-like growth factors, those are produced by the liver, and we know that it's a main center of metabolic type of activities, and this particular IGF1 and IGF2, these two factors are initiating, igniting reproductive type of system. Posterior pituitary, which has oxytocin, and it releases oxytocin. It releases oxytocin produced by the higher centers of the brain from hypothalamic region, and it is only stored for some time, and then it is released for its action. Oxytocin has important role for initiating Ferguson's reflex at the time of parturition. Violent type of uterine contractions are under the effect of oxytocin, and let down with milk reflex, that is the action. Next one. Next. Fine. Anterior pituitary, this is at the last. Here, these things we have already developed, discussed, that FSH, LH, they have important role in cyclicity, in cyclic animal reproduction. Prolactin is important. It is related with maternal sentiments, emotions, behavior. So everything, maternal instinct is related with this prolactin, and also this is important as far as initiation. Please remember, only for initiation of lactation, that's all. Then lactation is a continuous process, that is not under the prolactin, but prolactin hormones are always found to be higher when animal is under stress. During summer, you will find higher prolactin levels. During scarcity, whenever there is drought, prolactin levels in case of the animal, they will go high. So stress is not only related with ACTH, those are also related with the prolactin. Growth hormone, somatotropin, and ACTH, these are the other two non-reproductive type of the hormones. Next one. The next, hypothalamus we have discussed, and GnRH is the most important thing that we were interested to. Next one. So next, please. Right. Hypothalamic pituitary interrelationship is important as far as the reproduction is considered, or cyclicity is important. Hypothalamus will release GnRH through hypothalamic hypophyseal portal system. These factors, they will come to anterior pituitary. In turn, anterior pituitary will release FSH. This is the first and the foremost event. It is initiating from hypothalamus, and therefore hypothalamus must be sensitized with many factors, with health of the animal, with increasing body weight of the animal, with insulin like growth factors, with glucose level, with environment, with seasonal comfort, with managemental comfort, with nutritional comfort. And when those things they synchronize, hypothalamus has to release these type of factors. Once FSH comes into the bloodstream, it will reach to the ovary for the growth and the development of follicles. Friends, ovary is a stock of follicles. It is a stock of follicles. Primary follicles are abundant in the cortex of the ovary, and these follicles, they will get the FSH action. Total load of follicles in the cortical region of the ovary is nearly 1,25,000 with the right ovary and 1,25,000 in the left ovary. So whatever the stock is there, 2 lakh, 2.5 lakh, 3 lakh, everything is ready at the time of the birth, and that is available for the growth and the development. But irrespective of hormonal stimulus, friends, constant number of follicles are allowed to grow and develop as per a perfect time, or as per a unique time, at constant time. Say for example 10 days, and quantity say 10 follicles. So 10 follicles are allowed to grow and develop at the rate of 10 days. This is hypothetical, but this particular rate is constant. And remember, this particular rate is regular throughout the breeding life, whether the animal is pregnant, whether animal is postpartum phase, whether animal is in sexual rest, whether animal is in cyclicity. Every time 10 days, 10 follicles, they will grow and develop. They may not develop up to the mark, but they start development. This particular activity is a regular activity in the breeding life of female. This is important one. And this particular rate in terms of number of follicles and rate in terms of duration, after how many days the second cycle, third cycle should continue, it depends upon the species, but that is constant. The role of FSH is not on resting follicles. It is always on those which are allowed to grow and develop by the ovary, by the cortex, from the pool, from the total stock. Those which are lucky one, and they are in the phase of growth and the development, they are benefited by the FSH, and their growth and the development is under control of FSH. Such a type of 8 to 10 follicles, they grow and develop. And therefore, follicular development is in 3 phases. Number 1, recruitment. Number 2, selection. And number 3, dominance. Recruited follicles are 8 to 10. Selection. Out of these 8 to 10 developing Graafian follicles, only one is allowed to grow and develop toward the final maturity, and which develops to final maturity and pre-ovulatory stage is the dominant one. Recruitment, selection, and dominance. This is the sequence. Now, once this particular cycle is going on, the second part of FSH is played in the ovaries that under influence of this FSH, follicle develops, but this follicle secretes estrogens. Once estrogens they come into the picture, on one hand they will suppress FSH, and on other hand they will increase the level of the LH. Who is the hormone responsible for FSH LH balance or FSH LH control? Is estrogen. FSH will increase estrogen, but estrogen will decrease or it will inhibit the level of the FSH, whereas it will stimulate receptors for LH. More estrogen, more is the LH, and estrogens in this particular stage of dominance of the follicle produce signs of the stress. That is the stage of the stress. Once FSH level is low, estrogen is the hormone which is stimulating again to the hypothalamus for release of another GnRH signal, and in presence of estrogen, the signal which comes from hypothalamus will combine, will mix that type of masala which will lead to LH-like hormone. So initially it was FSH-like, alpha, beta subunits, they will change, and now with the recombination of the available parts, LH will come into the picture for final growth and development of the follicle, for ovulation, and also for formation of the corpus luteum. So this is the all neuroendocrine control of reproduction in case of the farm animals. Next one, please. Yes, hypothalamus pituitary interrelationship, very important one. Here we have to note that specific hypothalamic nuclei secrete releasing and release inhibiting type of the factors. Tonic release and phasic release. This is also important. It is not a continuous type of release. Small pulses and small tone, continuous tone, that is maintained in the secretion of the hormones. Next one. Next. Very important is the feedback. Feedback. One hormone will increase the level of another hormone, whereas this developed level of the hormone will increase for the another third one, but it will stop, inhibit the earlier one. So this all complex is called as a feedback mechanism. How stimulus is leads to a reaction on that particular hormone. Here you will find that this is for male type of animals, that how the continuous cycle is maintained for spermatogenesis and feedback mechanism. The next one we will see more important, we'll see for females. Please, next. Here it is a female. Now see, whenever there is a FSH, FSH will stimulate estrogen, estrogen will lead to LH, LH will form LH receptors, LH receptors, they will favor for progesterone, progesterone will continue its cycle for that particular phase of some 16, 17 days, and therefore when there is a empty type of uterus, progesterone will be withdrawn. That withdrawal is under the effect of PGF2 alpha. PGF2 alpha will reduce the level of the progesterone. Reducing levels of the progesterone will indicate to the hypothalamus that I am now reducing. So please start the next slide. It is progesterone. Withdrawal effect of progesterone. Please underline. Withdrawal effect of progesterone is the signal for next cycle. It is the signal for next cycle. This part is more important than other hormones and their feedback mechanism. Withdrawal effect of progesterone that leads to initiation of cyclicity, that maintains the cyclicity in case of farm animals. So this entire role, which favors in a positive manner, is a positive feedback, which favors for inhibition of the hormone is a negative feedback, and the cyclicity is maintained like this. Next one. The next one, yes, this is for milk let down reproduction. Just I showed you the slide for male reproduction. This is for stimulus to udder. The calf will stimulate, it will stimulate to neuroendocrine system, and then oxytocin will come down into the picture, and within 6 minutes, the letting down reflex will be there. And this is a complex type of mechanism. Every time this particular pathway needs to be completed for a release of oxytocin. So, let down reflex and its path mechanism. Next one. Here is the role of prostaglandins. Prostaglandins. See, the ovary and its corpus luteum is related with the maintenance of the pregnancy. I mean, CL is related with the maintenance of the pregnancy. Once there is no pregnancy, once there is no conceptus inside the uterus, endometrium remains healthy, and this healthy endometrium releases prostaglandins. Prostaglandins are carried from endometrium through uterine vein toward the corpus luteum. Vasoconstriction is one theory. It is one theory that due to vasoconstriction, the blood supply to corpus luteum itself is reduced, and therefore there is a lysis of the corpus luteum. And another important factor is that PGF2 alpha, progesterone levels will go down with the structural degradation of the corpus luteum. So, luteolysis will stimulate for the growth and the development of the new phase, and this cycle is more important in case of the animal for continuation of cyclicity. Had there been no prostaglandins, never ovarian corpus luteum would have recognized the availability or non-availability of pregnancy in the uterus. And that this mechanism, local hormones, prostaglandins, they are playing more important role. Next one. Prostaglandins are available in synthetic manner also. Pregma is available. Pregma D is also available. Dextrorotatory has a double effect than that of the pregma, and therefore pregma D, you must have seen this. So the type of compounds which are available with the prostaglandins, those are more important because their half-life matters. More effective is the role by synthetic type of the prostaglandins, and we are regularly using these type of prostaglandins in the clinical practice. So please remember that if it is luteolase, natural one, so 2 to 3 minutes half-life in the blood. If it is instrument, it is 500 microgram, it will remain 3 to 4 hours. The solving 15 milligram is the dose, and it will remain for 2 hours. Of course, other things are not available in our Indian market. So likewise, our judgment remains with the half-life of the prostaglandins for clinical use. Next one. Proestrus -2 -1 day, and this proestrus is characterized by high levels of FSH. High levels of FSH, developing Graafian follicles, palpable type of follicles on both the ovaries, but those are developing type of follicles. No signs of the estrus, but this particular phase is a preparatory stage, and animal prepares for expression of the estrus. This is most important, the highest level of FSH during the proestrus phase, -2 and -1. Next one. Estrus, we know that it is a behavior of estrogenic activity or expression of estrogenic activity. Dominant follicle must be available for expression of the estrus. Estrogenic activity is expressed by vascularity, muscularity, tonicity, and glandularity. Clinically, these are the 4 important things: vascularity, muscularity, tonicity, and glandularity. We can explain everything with this. Unless there is a developed Graafian follicle on the surface of the ovary, never recognize that particular stage as estrus 1. Signs may be like estrus, but follicle is mandatory. Follicle is mandatory, and therefore follicle, developed follicle has this role, and therefore we are getting 30 different types of estual signs in case of the animals. In case of estrus, the duration of the estrus is different in different breeds. How much is the duration of the estrus is the responsibility of owner to record. Owner is expected to record the duration of the estrus. Detection of the estrus is the responsibility of the owner, please remember, and therefore there are estrus detection aids, estrus detection kits, estrus detection equipments. Everything is related with the detection, and the detection part is part of owner and his management. Two things are important. Number one, estrus detection efficiency. How much owner is efficient for estrus detection? There are 100 cows which are cyclic one. If owner is detecting correctly with his AM, PM rule or whatever, maybe the electronic gadgets with him, he is detecting the estrus. His efficiency is that total number of animals in estrus and total number of animals he has detected. Out of 100, if he is detecting only 75, his estrus detection efficiency is 75%. He has lost 25 animals, even if they were in estrus. Estrus detection efficiency. Now the second, it is not that the owner should efficiently detect the estrus, but the estrus which is detected by the owner must be accurate, and that is estrus detection accuracy. What is accuracy? He has detected 75 animals in estrus, but only 70 were truly in estrus and 5 animals were not, but still he has recognized this in estrus. That is his accuracy. More close type of observations and 100% efficiency, 100% accuracy. This is required from the owners. Both the parts are related with the owners. Now, during this particular phase of the stress, animal is not under control. Please remember, clinically, when the cases are presented to us, animals are not under control. Mentally disturbed animals, they behave erratically during this particular phase. Only very calm and quiet animal will become restless. He has a very rash type of behavior with the owner during this particular period because animal is receptive. There is no other reason. Animal is receptive. Receptivity is characteristic with these stress. And highest level of estrogen that we have to understand. Basically, when we say stress has maximum estrogenic activity, that indicates it has lowest progesterone activity. Less than 1 nanogram progesterone is the estrol stage. Less than 1 nanogram is the estrol stage. And therefore, this part is very important one. Next one. Day 0, it is estrus. Mate estrus, very important one. During mate estrus, ovulation will occur. Developed Graafian follicle will occur after cessation of the estrus. 6 to 12 hours, 8 to 12 hours, 6 to 8 hours. This is the period after which animal will ovulate the Graafian follicle. This dominant pre-ovulatory follicle is ruptured, it bursts, and the bursting is caused by many lytic enzymes. They cause lysis of the wall of the follicle. Many lytic enzymes, lysosomes, are breaking the wall of the follicle, and then the ova is released. Ova, along with its cells, many cells, corona cells, that is released. It is not only a ova as a ball, but this ball is engulfed or it is covered by 400, 500 network of cells around the surface of the ova that is released along with the leaker follicular or follicular fluid. During metastasis, maximum LH, please remember, maximum LH. This LH is required for luteinization of the cells. Granulosa cells and the theca cells are converted to lutein cells. And once the cell becomes luteal, it produces progesterone. It produces estrogen. It's converted to testosterone, and testosterone is converted to estrogen. So such is to progesterone. So this conversion is most important, and this is by the enzymatic process. Once again, it is an enzymatic process. Metastasis is a short period of 2 days. Only healthy animals, they have metastasis bleeding, and that too in case of crossbred cows and HF cows. Our native cows are very, very less in number, but those who are which are healthy, definitely they will show metastasis type of bleeding. But this is not seen in case of buffaloes, please remember. And this the metastasis bleeding is also not seen in case of small ruminants. Characteristic only in case of healthy crossbred and HF type of cows, exceptionally in some well-grown, well-developed, well BSC carrying cows, native cows. During this luteal phase, during this phase that is metastasis phase, chances of fertilizations are there. Animal is in the phase of fertilization and post-fertilization event, and therefore this internal events they are going on during metastasis. The next one is the diastasis stage. Next one, and the diastasis stage is maximum stage. It is nearly 15 to 17 days. It's not 5. It's 15 to 17 days. And most important thing is that this predominant stage where CL is structurally and functionally developed up to day 7. From day 7 to day 14, it is a developed type of activity. And from 15th day onwards to 17th day onwards, it's a regressing type of activity. And therefore, please remember, palpation is most important one, and every professional graduate, please remember, every professional graduate is expected to palpate the CL as developing, developed, or regressing. Same thing is for follicles, developing or mature Graafian follicle. We have to identify these 5 things. Then clinically, nothing is required to treat infertility cases. If we follow a cycle of one particular animal for 21 days, our ideas will be clear, crystal clear ideas. And if we are able to palpate the ovarian activity, we are the only professionals who can treat any type of infertility case with the ovarian activity. So developing corpus luteum, developed corpus luteum, and regressing corpus luteum. Differentiation should be there, and we should go for regular palpation of these activities. In case of diastasis, progesterone level will increase up to 7 days, and therefore no need to give any type of progesterone from day 0 up to day 7. Please remember, let there be any case of infertility, there is no role of progesterone from day 0, that is from estrus up to day 7. From day 8 onwards, you can go for progesterone supplementation for checking early embryonic mortality, for continuation of the estrus and whatnot. 8th day, 12th day, 16th day, 8th day, 11th day, 14th day. You can have your own schedule and you can supplement progesterone from 8th day onwards. This particular stage is the most appropriate stage to hit the corpus luteum. From 8th day onwards, go for luteolysis. Whenever there is a subclinical endometritis, go for luteolysis. Everything will be cleared. Uterine contents will be clear. So mature corpus luteum will respond to luteolysis. Only mature corpus luteum will respond to luteolysis, otherwise not. In biological sense, only mature corpus luteum will respond to market available synthetic prostaglandins. That is most important. The next one. Next. Rate hormonal profiles during the estrus cycle. See, waves are shown, and these waves, they indicate that yes, follicles they are developing. Follicles they are developing. Levels they indicate that cyclicity, it is a regular issue, means as far as the follicular development is considered, it is a regular issue. And in all cases, this is all controlled by estrogen and progesterone. Estrogen level goes up, then it comes down immediately and it is converted into progesterone. Progesterone level goes up, it is maintained for some time, then it goes down. Once again, estrogen comes up. In this particular manner, this particular cycle, estrogen progesterone cycle, pedals of a cycle, those are playing the role. And when level of the estrogen and progesterone is constant, when pedals are constant and those are at equal level, animal is anestrus. When estrogen level goes up, progesterone level goes down, or when estrogen level goes down and progesterone level goes up, there is a pedaling, and this pedaling of the cycle indicates cyclicity. Remember this cycle pedal, as far as estrogen progesterone is considered. So this part is important one. Next one. Next. In the follicular phase, it is important that proestrus and estrus, these phases are completed. Follicular phase is for 4 to 5 days. Luteal phase is more, and it extends from 16 days to 16 days. Metastrus and diestrus, these are the events during the luteal type of phase. Next one. This is a table which will indicate us about the length of the cycle in case of farm animals. It will also indicate us about the time of ovulation and which is the time for artificial insemination. Friends, it is very, very important to note that the third phase of the estrus is the best time for artificial insemination, as far as cows and buffaloes are considered. Of course, in case of mares, alternate day service is required, and in case of sheep and goats, services are in the third phase of the estrus. Buck will cover the females only in the third phase. Next one. This is a story which starts with oogenesis. It turns to formation of a dominant follicle, ovulation to fertilization, and then conceptus. One follicle out of 8 to 10 selected follicles becomes a dominant follicle, pre-ovulatory follicle, and this follicle has important role to play for final maturity. And it's not only the role to grow himself as a final mature Graafian follicle, but it has to stop other growing follicles. That is also equally important. So this part is important one. And when we discuss about the Graafian follicle, centrally located ovum covered by layers of corona cells, this particular part is thrown out. It is expelled out from that of the ruptured Graafian follicle with ovulation. Next one. See, always it is important to note that it is not only the absolute level of the one particular hormone, it is the ratio of that particular hormone with its counterpart. Let it be FSH, let it be LH. It's a ratio is important. Estrogen, progesterone ratio is important because this ratio will be essential for production of receptor sites, which are important one for the role of the next expected hormone, next incoming hormone. So receptor formation has a role, and those receptors produced under the effect of ratio of those steroids or those protein hormone. So this type of important information is important one, because every time, even if the levels are high, we are not getting positive type of results clinically. It is only because of the total number of receptors available and total effect of that particular organ, target organ, which is responding to the action of the hormone. The next one. I have already told that there are something like 1,25,000 follicles at the time of birth. At the time of puberty, it starts recruiting the follicles, selecting the follicles, 8 to 10, like this, in case of cows and buffalo. In sheep and goats, the story is not so different, because once the stock of the follicles is exhausted, animal is senile. Animal is senile. That stage is rarely seen by the farmer breeders. But we are lucky enough that yes, there are animal owners who have 21 lactations from cows, 19 lactations from buffaloes, and let us salute this such a type of animal owner, because they are not putting their animals for culling. I will not say killing, culling at least. Prestigious black gold of our country, buffaloes, are unnecessarily being slaughtered after 5 or 6 lactations. It is only because farmer breeders, they have poor knowledge of animal reproduction, and they are not controlling the reproduction in case of the animals. When we are there to make the animal cyclic and pregnant, putting the animal to the slaughter is not expected in case of buffaloes. Let there be pressure. I will say pressure, because buffaloes are taken up from the farm under pressure that now 5 lactations are over, put that animal to slaughter. There is no ban on buffalo slaughter. Why not to put the buffalo on slaughter? Very sorry and very pitiable type of the situation. So this type of important information, that follicles, as long as those are there in the cortical region of the ovary, animal will come into the stress. Now these type of lifetime calvings, lifetime pregnancies must be considered in case of animal reproduction. Next one. The next one, right? Types of ovulations, very important one for we clinicians. It is always a spontaneous type of ovulation in case of cows, buffalo, sheep, and goat. After cessation of the estrus, maximum estrogen, but this estrogen will initiate, it will stimulate release of LH, and LH has 3 roles. Number one, behavioral signs of the estrus are expressed under effect of LH. So it is a estrogen LH combined relationship. In Maharashtra, we can say Yuti or Agadi, estrogen LH type of group will go for expression of the estrus. This is number one. Number two, it will go for ovulation. LH will bring about the ovulation, and LH will bring about luteinization of ruptured Graafian polygon. So that is the ovulation, and that ovulation is a normal type of event in case of the animals. Next one. Next. Whenever there is ovulation, we expect that for better chances of fertilization, there must have been availability of sperms in the reproductive tract of the female. Ovulation 6 to 8 hours after cessation of the estrus. Insemination at the last phase of the estrus. Means we are 12 hours before, as far as the AI is considered. 12 hours before. And 12 hours to 24 hours is the time for capacitation of the sperms. It is time for capacitation of the sperms. Remember, on insemination, the total number of 10 to 12 million live, motile, normal sperms are placed into the reproductive tract of the female. These sperms are stored in the bunkers, mycellae, bunkers of cervix, mycellae of the cervix, where the batches of the sperms are retained for a period of 18 hours to 24 hours, and a batch, another batch, third batch, fourth batch is released toward the uterus. Timely release of the sperms is a function of the cervix. So live and motile sperms are released toward the uterus, toward the bifurcation point, toward the uterine horns for fertilization process, whereas all dead sperms are removed through the discharge toward the vaginal side. And therefore, selective absorption of the sperms is a function of the cervix. That is important function of the cervix. And this particular capacitation process starts with cervical mucus. It starts with uterine mucus. It starts with oviductal mucus. And slowly these sperms, they enter into the fallopian tubes. 4 minutes are required. 4 minutes are required for transport of sperm from cervix to fallopian tubes. 4 minutes. At that time, if the ovum is not available or if it is not ready for this type of fertilization, that batch is waste. Second batch, third batch, and therefore we are placing 10 to 12 million sperms. How many sperms are required for one single successful fertilization? Is 3,000, 4,000. That's all. In case of cows, buffaloes, even in case of mare, 3,000 to 4,000 live motile sperms are required at the site of fertilization. How much we are placing? 10 to 12 million live motile and normal sperms. So this movement, it will carry the sperms toward the fallopian tubes in the ampullary region, and then there is the fertilization process. This process is also important one. This should take place in all circumstances. Even if we expect normal fertilization process, it is not possible to monitor this particular process externally. We are only expecting the things and the changes which we are expecting for fertilization. The next one. Next, please. That's right. Capacitation. This is a expected time for capacitation. It may be little more. Sperm will undergo the process of capacitation with a different type of process. Capacity to become ready for fertilization. And this particular capacity is given up, or it is provided to the sperms by different steps of capacitation process. Whatever the lysosomes which are available in the head of the sperms, those sperms, they play a very important role as far as their penetration into ovum is considered. It is penetration into ovum is considered. Remember, ovum, the ball, is covered by, say, for example, 500, 600 corona cells tightly packed on the ball of ovum. Each cell is fixed with each other by a cementing substance. A cement is available in between the bricks, and the bricks are corona cells. Hyaluronic acid is the cementing substance. Now, breaking of this cement is necessary with the help of enzyme, and the enzyme is hyaluronidase enzyme. Hyaluronidase enzyme will break these walls, brick walls, corona walls. So each cell will get separated out. Why 3,000 sperms are required at the site? They are required for the concentration of the enzyme, hyaluronidase. And once there is a concentration of the enzyme, then only separation of corona cells is possible. Otherwise, if there are, say, 200 sperms at the site of fertilization, there will not be fertilization because ovum will be dying in the layers of the corona cells because those are not getting separated out. Those are not getting separated out. So this is the basic thing at the site of fertilization. Next one. All right. The sperm is more important as far as its head region is considered. Outer acrosomal layer and internal acrosomal layer, they unite first. Acrosomal cap will release the lysosomes, RDX-like material for bursting the zona pellucida. So this particular lytic process is also important. And here in the figure, you will find that the centrally located ovum covered by coronary cells. This separation of all cells is necessary. It is not that some cells are remaining there. No, everyone should go out. Then sperm will enter in perivitelline space first. It will enter in perivitelline space and it will take rounds and rounds. Once the sperm is in the perivitelline space, there is a vitelline block or zona reaction. If it touches to the wall of the ovum obliquely, then only the sperm is engulfed. Immediately, sperm is engulfed by the ovum. The nuclear part of the sperm head is taken up, and tail region will be allowed to flow, to rotate in the vitellus. This particular part is important in the process of fertilization. Next one. See, this is the something like steps in the fertilization process. Sperm and the ovum, they unite with pronuclei, and union of male and ovum pronuclei, that leads to fertilization. That is the part of fertilization. Once there is establishment of 2n number of chromosomes with the ovum, there is a stimulus to the entire cell for the growth and the development, and therefore cleavage starts. Why cleavage starts? It is because of the stimulus of union of male and female pronuclei. Doubling will start immediately once the male factor is in, and this male factor is responsible. It is stimulating the cleavage part, 2 to 4, 4 to 8, 8 to 16, and then process will continue throughout the pregnancy, and then there is a formation of a conceptus, which is recognized by the endometrium, which is further recognized as embryo by the uterus, and the pregnancy will continue. In this particular process, whatever expression of foreign proteins through conceptus is received by the female, that needs to be stopped. Female should not recognize that yes, some foreign proteins are there in the body. That immunosuppression is brought about, and this immunosuppression is responsible for maternal recognition, for implantation, and for continuation of pregnancy in the females. So friends, it is important that this conception is a very complicated type of process. Clinically, we should be perfect in synchronization, ovulation, and artificial insemination. After artificial insemination, it is our bounden duty. It is our bounden duty to ask farmer bidders that please check, number one, how much time this animal continues estrus after I have inseminated that animal. This is number one. And number two, please check that this particular animal express the subsequent estrus after how many days, or it stops the cyclicity after 21 days. So these are the two important instructions which must be passed to the animal owner. As per the conception is considered. Next one. I have few slides for artificial insemination also. This is something continuation of the fertilization process. Syngamy and other abnormalities of the fertilizations are generally stopped, and only single sperm is allowed to fertilize the ovum. So polygamy, polyspermy is not allowed in the fertilization process, and such a type of conceptus, they will die immediately. There is no growth and the development of such a type of conceptus. Next one. Next. This is the related with something like parthenogenesis. Why there is parthenogenesis? Why there is artificial activation of ova? That is possible in experiments, but normally only normal fertilization will continue in pregnancy. Out of 100 normal cyclic cows, 80 to 85% cows have normal fertilization. 15% may have failure of fertilization. But normal cyclic cow, they record 80 to 85% normal fertilization rates. They have 10% embryonic mortalities and 75% conceptions, embryonic developments are there. So out of 100, 75 animals are expected to become pregnant in normal case. In case of repeat breeders, we are going to discuss this in the next week. But in normal course, 10% embryonic losses, 15% fertilization failures are expected in normal cases, where there is no justification and there is no ideal type of incident where that all normal 100% cows will have 100% fertilization and 100% pregnancy. This will not possible in reproduction because there are many hindrances. Right. Next one. Coordination of events for fertilization to occur and time of breeding. I have already given important things about this, that how much time is required and why there should be synchrony of ovulation and artificial insemination. Next one. Next. This is also a continuation of the same. Next one. Next. About artificial insemination, one should take reasonable care for thawing. 37 degrees centigrade for 30 seconds is a standard one. Straw should be completely dipped into the lukewarm water. After 30 seconds, straw needs to be cleaned with tissue paper. Loading of the gun should be proper. This particular process is always to be attempted only after confirmation of estrus. Please remember, no thawing is expected when animal is presented to the clinics. First, examine the animal. Confirm that animal is in estrus with number 1, open cervix, tonicity of uterine horns, and follicle on one of the ovaries. These are the only 3 pathognomonic perfect characteristics of the estrus. A veterinarian is a judge and he will confirm the estrus only on 3 points: open cervix, tonicity of horns, and developed Graafian follicle on one of the ovaries. This must be the finding. This is confirmed. That is our duty. After that, he will have to clean his hands. He will have to go forth. Not that he first is going for drawing. He has confirmed the estrus and immediately he is going for artificial. That is not expected. First confirm the estrus. Many times, when efficacy and accuracy of heat detection is very poor in case of Indian farmers, we should not lose the straw. When we are going to use semen, we should never do such a type of the things. That is really important, right? Another important thing is that this particular thawing process is more crucial than freezing. Thawing will lead to double losses. Freezing will lead to losses, but double than that, those are taking place at the time of thawing, and therefore thawing must be crucial and it should be followed scrupulously. Next one. Yes. whenever we are inseminating the animal, it is mandatory. It is mandatory that there should have been an assistant to dilate the lips of the animal so that we can pass our gun without touching to the vulval membrane, vaginal membranes, and directly into the cervix. Please remember, no one should say that, yes, I'm so perfect that I can myself inseminate a cow. No need of any assistant. No. Owner, assistant, attendant is required for dilatation of vulval lips. Gun should move without disturbing, without touching the side walls of the vaginal mucosa. It should directly reach to the cervix. That much skill is important one as far as the artificial insemination is considered, and mid-cervical insemination is recommended. It should not be followed by everybody. If you have different observation, you can follow your own. In case of insemination, there is no standard type of recommendation about the time and the site. Number of sperms are constant, but time and site, you can have your own diagnosis, your skill. Recommendation is like this. The next one. That's right. Once the cervix is located, that gun should glide on the ventral vaginal wall for stimulating to the cervix. This is a normal course. Gliding action, gliding type of the withdrawal is necessary. This is number one. Massage to the vulval lips, that is also important one. Prefer that animals should move some distance after insemination. That is also important. That during walking, animal will not contract much, and animal will not void out the insemination drop. So that is for that purpose. If animal is allowed to walk down, definitely that will also help. So these things are very, very important one. And I think with the proper insemination procedures, we will succeed in getting the fertilization. Next one. Same thing. Next. So thank you. I think I have started at 10, and now it is nearly. We have started at 9:10, and it is 10:50 in my watch. My mobile numbers you will be getting like this: this 8237682141, 8237682141, or 94226 57251, 94226 57251. So you are free to have open professional dialogue at any time, right now and even afterwards also. So thank you, State Gazetted Officers Union and Veterina, for providing me this opportunity to interact with you. I'm a little more interested in clinical type of the things, and therefore basics and this PPT, everything is for you. Let us use it. You can add it, and you can improve whatever you would like to add in it. So likewise, that will be a useful thing for you. Thank you very much.
Thank you, sir. I
have been asked from Dr. Ajayji that what type of remedies we can use for irregular type of the stress. Irregular stress is that stress which is not expressed at the interval of 21 days. Animal is in stress on second day, third day, fourth day, fifth day, 10th day, 8th day, 12th day, 17th day. It is all irregular. 24th day, 35th day, 37th day. This is all irregular. So if the time interval between the two successive consecutive cycles, menstrual stages is not 21 days, then it is irregular cycle. Irregular cycle indicates that there is some pathology. Irregular cycle which has time lapse, time duration less than 21 days, It is indicative of uterine pathology. It is indicative of uterine pathology. When the cycle length is more than 21 days, it has pathology of embryonic deaths, embryonic mortality. So accordingly, we can assess this type of stress. But in anyhow, if animal is not cycling regularly, animal is not fit for insemination. In normal course, this is our rule. Those animals which are exhibiting stress after 21 days, they are fit for insemination. Insemination technology has never said that animals which are in stress are fit for insemination. No. Animals are always in stress. Animals, they have tendency to exhibit this stress. Animals, they have tendency to exhibit even during gestation. That doesn't mean technology has asked us to inseminate. Inseminate to regular stress is the technology. Inseminate to and regular stress is that stress which is expressed by the animal after 21 days. And who will check this? Animal cannot check whether this is a regular stress or irregular stress. Owner will have to check it. So he will have to record it that on 1st of February, animal was in stress. Today is 21st of February. Today animal is in stress. First stress is not fit for insemination. 1st February, 21st February is fit for insemination. Dr. Arif Sheikh has also asked that, can we expect a sort of Arif ji, chat. Yes, I will go to chat and then I will come to you. Arif ji, can you ask me this question by just?
Yes, Dr. Arif. Sir, you are requested to. Okay.
Please. Dr. Sir, please.
Sir.
Actually, I was.
Can we expect bloody discharge after successful insemination?
Yeah, we can expect. It is a normal physiological. The bleeding which occurs is a metastas type of bleeding. It has nothing to do with the professional skill of a veterinarian. I am confident and sure about all my professional brothers and sisters that they will never injure any part of the reproductive system. If the blood is observed after insemination, it is a metastas type of bleeding. It is 24 hours to 72 hours. Time lapse is 24 to 72 hours after insemination. Owner has to say, and owner will say that you have inseminated and I have seen the blood. It is not like that. That blood is normal, physiological. It's only a bursting of veins which are superficial one because of the higher blood pressure, and the blood clot which comes into the endometrium is slowly discharged out. Whatever time taken is the duration of metastasis. By that time, sperms are in the fallopian tube. Conception is there in the fallopian tubes. It is safe for 5 days. By that time, endometrium is healthy, and we are not at all putting any type of injury to the animal during insemination. 100% true.
Sir, does it affect pregnancy?
No. It has no role. Metastasis bleeding has no role. It is only a physiological process. It happens only because of excess blood pressure in the endometrium. Those vessels which cannot sustain the blood vessels, superficial blood vessels, they rupture, they burst out. The clot which is formed inside the endometrium, it takes little time to come out, and therefore we observe it after 24 to 48 hours. I said that this particular bleeding is only after insemination. It is only after cessation of the stress. It is only after insemination and 24 hours afterwards. By that time, all the sperms from cervix, they will definitely go to fallopian tubes, and there is a possibility of successful fertilization.
Because sir, farmer always think that if bloody discharge come after insemination, there will be no pregnancy. So I ask this question.
You are very correct. It is see, anybody, a layman can have this type of attitude that yesterday you have passed the gun. That was a very small type of pinching structure that must have injured mucosa. This will not happen. We are handling genitalia in a very gentle manner. Unless we hold the cervix, we are not passing the gun. The tip of the gun, we are observing, we are monitoring, and we never penetrate anything. If there is any obstruction, immediately we try to withdraw the gun to some extent, and then we observe that the course of the gun, ascending course of the gun is smooth. That we always follow.
Thank you, sir.
Yes.
Sir, can we inseminate after metastral bleeding, or up to how much hours we can inseminate after bleeding?
Insemination is always in the last phase of the estrus. We are leaving that particular animal by asking the owner to observe the animal in. I'm sorry, there was the connectivity problem. See, doctor, once we are inseminating the animals, after 6 to 8 hours, animal should go for cessation of the estrus. Animal should stop the estrus. But if an animal continues the estrus beyond 12 hours of our insemination, so re-insemination is always after 24 hours. It is after 24 hours of first insemination. So please remember, never repeat insemination before 24 hours. We have to inseminate after 24 hours only.
If there are no more questions, sir, there is one more question from Dr. Kalidas, that in some heapers like gear, cervicitis, heart swelling of cervix seen, what is the remedy? But I feel our questions are related to pathology of reproduction. So we are going to cover these topics in a next lecture, which will be taken by a respected sir only. So, sir, in that series, sir will be talking on the infertility causes and its treatment. So I think all these problems, all these questions can be taken in the next session already. And with the experience of this lecture, I feel that for infertility we may need two separate lectures of this, sir, so that we can cover it in detailed manner. Today, sir has been talking for almost 2 hours, so we are really thankful, and most of us got an opportunity to refresh the basics after almost 15-20 years, and for some people it was for almost 30 years. So we are so much thankful to respected sir for giving as usual a marathon talk on the ruminant reproduction. I am also thankful to all the participants who have been there since last 2 hours in this meeting. So thank you one and all, and good night.